Hey everybody, so we're finishing up with this lecture here that's basically our digestive system notes proper, our mainstay packet, the digestive system, chapter 23, as it's called at the very top here. And we're going to finish up with the two largest accessory organs of the digestive system, the liver and the pancreas. So I'm quickly going to refer back to our PowerPoint that the publisher provided us with some better pictures, just because these pictures have been photocopied way too many times. But first, I just want to use this as a springboard to talk about the basic gross anatomy of the liver. Liver is broken up into four different lobes: the right lobe, left lobe, caudate lobe, and quadrated or quadrate lobe, as it's listed in the book. The liver suspends from the roof of the abdominal pelvic cavity. So the abdominal pelvic cavity is completely surrounded in peritoneum. That's needless to say, right? That's the serous membrane that envelops the entire outer boundaries of the abdominal pelvic cavity. Well, at the roof of the abdominal pelvic cavity, just inferior to the diaphragm, that parietal peritoneum folds in on itself to form a two-layer sheet. It's basically a mesentery known as the falciform ligament. The falciform ligament, let me write this out, falciform ligament then basically suspends and holds in place the liver. So it suspends the liver from the diaphragm, essentially, via the parietal peritoneum folding in on itself to form visceral peritoneum. And then also that same visceral peritoneum then separates as the falciform ligament connects to the liver and then completely enshrouds the liver and helps to hold it into its place. Okay, so now let's look at the gross anatomy that I just explained to you on the PowerPoint. So now we're looking at an anterior view of the liver. This is the view of the liver that you're probably most accustomed to seeing, especially when you're looking at an anterior view of all of the organs within the abdominal pelvic cavity. Right here, we have the large right lobe, the largest lobe of the liver. And then over here on the left, we have our left lobe, also a large lobe, but not as large as the right lobe. And then in between the two lobes, let me switch to a different color. I don't want to get out of here. I want to switch to a different color. Okay, let's do blue, nice complementary color. Here you can see the falciform ligament between these. So again, the falciform ligament basically helps suspend the liver from the roof of the abdominal pelvic cavity, which is determined by, or I should say the best way, reinforced by the diaphragm, which is just above it. So also you can see the gallbladder tucked here behind the right lobe of the liver. This is our gallbladder, very appropriately colored in green. So now let's look at the back of the liver. And really what we're looking at is kind of the posterior inferior view of the liver. This is where we can see the other two lobes of the liver. You can't see those other two lobes, the caudate lobe and the quadrate or quadrated lobe, from the anterior view of the liver. So now let's move, and now we're looking at the same gross specimen from the back. So let's highlight those same lobes that we talked about. So right here, you can see our right lobe, and then over here we can see our left lobe. And then between these two large lobes, we can see our other two lobes. This right here is going to be our caudate lobe, this more superior lobe. And then this more inferior lobe is going to be our quadrate lobe. So the root word in caudate is caudal, which means tail. So this lobe, I guess the anatomist thought looked like a tail. I don't think it really looks like a tail. It does have tapered, or it has a tapered end to it. I don't necessarily mean that qualifies it as looking like a tail, but that's what they named it. The quadrate lobe has a name that's a little more sensible. It's roughly the shape of a rectangle, right? So the root word here, quad, gives reference to the four roughly straight sides of this rectangular lobe. I want to say square because that would seem even more appropriate, but it's not square. It's rectangular. So we can't make it something it's not. But that's where we derive this term quad from. It refers to its four relatively straight sides. Okay, so here we have the artist diagram where we can see these same structures. So here we have the right lobe we just gave reference to. And then here we have the left lobe. And then here we have our caudate lobe with the tapered end. And then here we have our roughly rectangular quadrate lobe or quadrated lobe. And then of course here we have our green gallbladder. All right, so the most fundamental, independently operating, all-encompassing subunit within the liver is actually the lobule, which can only be appreciated on a microscopic level. So you see this little box right here? Let me switch colors. And I don't want to go back. I want to switch colors. I don't want to end recording. I want to switch colors. Well, that's the problem, is that Mac towards kind of a control system where you just use the gestures, but the software doesn't really recognize the gestures very well yet. So I feel like, you know, it would be great if we had software that was really good at just recognizing gestures, but we really haven't gotten to that point. So honestly, I would rather just have buttons and get to that point where the gestures actually reliably work. Let's just use buttons. Anyways, so let me draw this in for you with black. So again, we're just taking this little area of the liver, overlying the cartoon, outlined by this black box, and we're blowing it up to see what's inside on a more microscopic level. And you can see all of these repeating lobules, these roughly hexagonal shaped subunits. So within each of these lobules, we have all of the self-contained functions of the liver in and of itself. So the liver on a gross scale is really giant homogeneous gland. What makes the liver really thick are all these individual microscopic lobules. So now we're going to talk about the different functions of the liver and the different architectural features of the lobules on our notes, and then we're going to go back and we're going to look at those functions and architectural features in action and how they work together, how it all comes together to create this greater picture on the diagram that's important. So we're headed back to the notes. Okay. So again, each lobe of the liver is separated into lobules, these little microscopic subunits. And the lobules have many, many hepatocytes in them. And the hepatocytes are the main workhorse cell of the liver. These are the cells that are actually carrying out all of the basic functions of the liver. Other structures that you see that are quite obvious, especially towards the center of the lobule, are the sinusoid, central vein, and bile, that is, canaliculi. So the sinusoids are like these little lymphatic vessel-like crevices which contain hepatic macrophages, which are otherwise known as Kupffer cells. And basically, like macrophages throughout the body, these Kupffer cells just engulf debris or bacteria or other pathogens that find their way into the liver. Okay, so right now we're just giving a shout out to the central vein and the bile canaliculi. I think that I'm going to wait until we get to the pictures to actually explain to you how they actually operate and fit into the greater whole. At the perimeter, or the 6 points at the edges of these hepatic lobules, we have 3 vessels arranged as a collective structure known as the portal triad. So let me draw a little lobule for you. At the corner of each of these lobules, you have a portal triad. And the portal triad, wait, what's happening here? I'm trying to get back to my marker. Oh, that's right, it's down here. Okay. So at the portal triad, you are going to have a bile duct. Come on, let's go. You're going to have a bile duct. You're going to have a branch of the portal venule. Change colors here. And you're going to have a branch of the portal arterial, and I'll explain the relevance of those coming up very quickly. So you see how I'm drawing in these little features here at the corner of this little lobule. And so that's going to repeat at all of these 6 points at the perimeter of each lobule. So again, this is just one portal triad right here, which contains a bile duct, a branch of the portal venule, and a branch of the portal arterial. Again, we're going to go into the relevance of that coming up shortly. Just want to make sure we get the language down first. So again, at each portal triad, we're going to have a branch of the portal arterial, and a branch of the portal venule. So the big picture functions that we have of the liver and of which are occurring in all of these individual lobules are processing nutrient-rich blood. So we're taking the nutrients, especially like the lipids, etc., and we're doing different chemical processes on them so that they're better suited for the body or the use of the body. We're producing plasma proteins, the most common plasma protein of which is albumin. You guys remember albumin when we talked about the blood. It produces bile, which is the main excretory product of the liver. And we're going to talk more about bile when we talk about GI physiology in lab. It detoxifies the blood, so it has a whole bunch of different specific enzymes contained within the hepatocytes that have a role in breaking down a myriad of different types of toxins which may threaten the body. It also works to help store glucose as glycogen. You guys remember this from AP1, right? Can also store vitamins and iron. All of these functions, A through F, are actually carried out by the hepatocytes, which are the main workhorse cells of the liver. And I'll point them out to you when we get to the diagram of the lobule. Then finally, the phagocytosis is going to be the role of the Kupffer cells, which hang out in these lymphatic vessel-like crevices called the sinusoids. All right. Okay. So right here in this diagram, these are the terms we want you to know for 1, 2, and 3. But the problem is the diagram is so bad that, I know a lot of people like to fill in the blanks correctly on their notes because it makes them feel better. But I apologize for this. We need to actually work on getting some color reproductions of these notes. Oh, I'm sorry, guys. This is just a little email that just popped up here. So again, it's filled in for you, but I think the greater benefit is to actually look at the diagrams in the book, which I'm going to show you through the PowerPoint right now, and you'll be able to identify all of these same structures that we're going over. So that's what I'm going to do right now. Okay, so here is the more technical cartoon version of one of these lobules. So again, at the perimeter of the lobules, you have the portal triad. So here's the portal triad right here. And you can see the portal triad is composed of a bile duct, a portal venule, and a portal arterial. And really, they're branches thereof, if you want to get really technical about it. So let's just start with this particular portal triad at this corner and go over its different components and how they contribute to the whole. So this branch of the portal arterial basically is a derivative of our common hepatic artery. So you guys remember the common hepatic artery or just hepatic artery? This is the artery which is delivering fresh oxygenated blood to the liver, which we learned about when we talked about the blood vessels. So it's a derivative of the hepatic artery. And why do we have these branches of the hepatic artery carrying fresh oxygenated blood? Well, it turns out that the liver likes fresh oxygenated blood, just like every other organ in our body, and that blood is delivered into the capillaries, and you can see that going on with these white arrows right here. So you can see the blood being delivered into these capillaries. Actually, not that one right there. That's coming from one of the portal venules. So let me erase that. Oops, not that one. But this white arrow right here, let me highlight it again. Let's highlight it with orange. This little arrow right here is symbolizing the fact that the blood is coming into these capillary beds surrounding hepatocytes. Hepatocytes are all of these kind of like light copper colored cells, by the way. So you have capillaries surrounding these cells, and so the fresh oxygenated blood is entering these capillaries so that the oxygen in this blood can serve all of these hepatocytes. And that's the end of the story. That's it. So you guys remember that we have this hepatic portal vein, which is collecting venous blood from our gut. And so as our gut is absorbing various nutrients, those nutrients get redirected into the venules that ultimately converge to form our hepatic portal vein. And this nutrient-rich blood through the hepatic portal vein is then routed towards the liver. So when the hepatic portal vein gets to the liver, and you can see it right here, hepatic portal vein, when the hepatic portal vein gets to the liver, it splits and 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 splits. Much like an artery would split, but in this case, it's actually a vein. And finally, it splits to the level that it gives rise to these branches of portal venules. And these branches of portal venules then service the 6 sides of each of these microscopic lobules. So basically what we have is venous nutrient-rich blood. Venous nutrient-rich blood that's being carried by these portal venules or branches thereof. So let's highlight one of these portal venules because this is really important. Let me get out the black. So again, here's one of these branches of the portal venules. All right. And so now we have venous blood being delivered into the sinusoids. And the sinusoids are these deep crevices that this venous blood travels through. And as the venous blood travels through, it passes along the side of the hepatocytes, and then the hepatocytes basically detoxify the toxins in the blood. They pick up the nutrients and process the nutrients. They pick up the glucose and they store it as glycogen. And so basically it's just this almost like filtration system where as the blood comes through, these hepatocytes are working on the blood as it passes along through the sinusoids. And the direction in which the blood is passing is towards the center. And eventually all of these sinusoids carrying this nutrient-rich blood, ultimately that was drained from our gut, empty out into the central vein in the middle of the liver. So each of these, or in the middle of the lobule rather, so each of these lobules has, I should say, a central vein. So that's the big picture. And then eventually these central veins are going to converge and converge and converge and converge and converge as any good vein does. And finally they converge into the very large hepatic vein, which is actually multiple veins, which then take that now processed, detoxified blood back to the inferior vena cava. So now that blood is suitable for circulation within our systemic circulation. So at the same time, between the hepatocytes, we also have these very, very thin vessels called the bile canaliculi, which I'm going to highlight for you in green. So you see this little webwork of vessels right here, the bile canaliculi. The hepatocytes, while they're processing the blood, are also making bile, and they deposit that bile into the bile canaliculi. And that bile is actually moving in the opposite direction as the venous blood. So we said the venous blood that comes in through the portal venules is headed towards the center of the lobule, right, towards the central vein. Well, the bile that's being deposited in the bile canaliculi, which is basically wedged between the hepatocytes, is moving in the opposite direction. The bile canaliculi are taking the bile towards the perimeter of each lobule, where that bile is deposited into each of the bile ducts. And then what happens is the bile ducts all converge, and then those bile ducts converge, and then those bile ducts converge, and then those bile ducts converge, and there's a lot of converging of bile ducts. And then finally, those bile ducts, trying to show you the biliary tree here in the PowerPoint presentation. Yeah, so here we go. These bile ducts basically find their way into the two largest bile carrying vessels within the liver itself, your right and left hepatic ducts. So here you see your right and left hepatic ducts. And then they converge to form your common hepatic duct. So here you have your common hepatic duct. All of these vessels carrying bile, just like the bile canaliculi and the bile ducts within the liver. And then what happens is we have this very unusual branch point where the common hepatic duct then gives rise to the cystic duct, which is a special conduit for carrying bile for storage, temporary storage in the gallbladder. And so that's what the gallbladder ends up being. It's just a temporary storage unit for this bile until it's needed. And by the way, this is a spoiler alert. What bile does is it helps physically break down fat droplets. It takes big fat droplets and breaks them down into small fat droplets. That's what physical digestion is. We went over physical digestion at the very beginning of this lecture, and you're going to talk more about it in lab. So this gets a little bit more complex, but it really isn't any more complex than it needs to be, or at least it isn't any more complex in the way I present it here. What happens is the bile finds its way down the common hepatic duct, and at this branch point between the cystic duct, which takes the bile to the gallbladder, and the common hepatic duct, we have a new duct formed called the common bile duct. This is the common bile duct that I'm highlighting in green for you. And so the bile finds its way all the way down to the bottom of the inferior most extent of the common bile duct. But then it's blocked by this little sphincter right here called the hepatopancreatic sphincter. So now it has no other choice but to start to back up in the pipe. And then as it backs up, it then heads backwards into the cystic duct so that it may be stored in the gallbladder. Does that make sense to everybody? It's not intuitive, but remember, this is no more complex than just plumbing. This is just plumbing. So think about it. If you have a sewage stack in your basement and it's plugged up, where's the sewage going to go? It's going to go back up in the opposite direction. And so it goes back up in the opposite direction and then fills up the gallbladder via the cystic duct. Now, when you just had a big fatty meal, this results in this sphincter, the hepatopancreatic sphincter, relaxing and your gallbladder contracting, and then the bile goes back out the cystic duct and then down to this branch point again between the common hepatic duct and the cystic duct to give way to the common bile duct. And then the bile goes back down the common bile duct and then it gets squirted out through the major duodenal papilla, this little mound with a hole in it, within the inner lining of your duodenum. So the bile gets squirted out into your duodenum, where it aids in the physical breakdown of fat droplets, aka the emulsification of fat droplets. So that's the big story there about what the bile is for and how it ultimately gets to your duodenum. All right, so let's go back. Again, this thing doesn't understand that I don't want to make marks. I want to move. I don't want to make marks. I'm trying to swipe this with my finger, so this, you know, these computers are not as smart as we think they are. I don't understand. It's like it gets blocked, and it thinks that it won't let me just break out of there. Okay, so now it's letting me move it. Maybe that's what I have to do. I just need to get out of it and then go back into it again. All right, so let's go back to where we were. So again, we covered all of these terms and their meanings. These diagrams are basically just little cutouts of the same diagram we went over. So let's talk about the gallbladder very quickly and the pancreas, and then we'll talk about these final diagrams here and then how it all fits together. So the gallbladder, again, is going to be this bladder we use for the temporary storage of bile. Like I said at the very beginning, gall means bile, so the gallbladder literally translates to the bile bladder. So the inner lining of the gallbladder contains these ridges called rugae, just like the stomach's inner lining contains the rugae. You'll see that when we do the urinary system, our urinary bladder has inner folds called rugae. And again, the rugae are always there just to allow for, facilitate the further distension of this organ. And the major function of the gallbladder, as we said before, is the temporary storage of bile. All right, so let's talk about the pancreas. Located posterior and inferior to the stomach, contains these little microscopic clusters of cells called acini. It's basically glandular epithelium that secretes pancreatic juices, and the pancreatic juices contain enzymes which are used to digest both lipids and proteins. You also do have sodium bicarbonate as well, which helps to neutralize the small intestine because the chyme that it receives from the stomach is very acidic. There's also water and salts within the pancreatic juices as well. So now let's put everything together and talk about how the pancreatic juices get to the duodenum and how the bile gets to the duodenum and how that all fits into this whole branching tree of bile-carrying ducts known as the biliary tree. So number 3 right here refers to the right and left lobes of the liver. And then number 2 is going to be the falciform ligament, right? And number 1, of course, is going to be our gallbladder. Number 4 is going to be the pancreas. And then this is the duodenum, this whole structure right here. Let me get a different color to label the duodenum. Let's do it in orange. So the C-shaped tubular organ is our duodenum, the first part of our small intestine, right? So again, let's review how this works as far as the bile is concerned, how the bile gets to the duodenum. The bile is going to be collected by the bile ducts at your millions, rather the edges at the millions and millions of lobules that you have within the liver. And then those bile ducts are going to converge and converge and converge and converge and converge. And they're finally going to converge until they converge into your right and left hepatic ducts. So here you have your right and left hepatic ducts. This is going to be your right hepatic duct right here, and this is going to be your left hepatic duct. Your right and left hepatic ducts are going to converge together to form your common bile duct. And then your common bile duct ends right about here, where it gives rise to this long duct called the common bile duct. Not the common hepatic duct. This is the common hepatic duct. All right? And then right about here, at this point where the common hepatic duct ends and then gives rise to your cystic duct, which takes the bile to the gallbladder, it then also gives rise or branches into the common bile duct. So this is the common bile duct that I'm coloring in right here. Again, let me label in the cystic duct for you. The word cystic, by the way, has the root word cyst in it, which basically means bladder. So the word cystic refers to the fact that the cystic duct is taking bile to the bile bladder, which is your gallbladder. And the common bile duct leads all the way down to the duodenum. So the story goes is that bile starts to collect into your right and left hepatic ducts, which converge to form your common hepatic duct. So the bile is deposited into your common hepatic duct. And then that bile keeps on going down into your common bile duct until it gets all the way down to the level of the duodenum. But remember, we have a little sphincter right here, the hepatopancreatic sphincter, also known as your sphincter of Oddi, by the way, that's constricted, and that blocks the bile from moving any further. So just like a plugged up sewer stack, it goes back in the opposite direction. And when it goes back in the opposite direction, it then goes up through the cystic duct and into your gallbladder for temporary storage. Then when you eat a nice big fatty meal, that hepatopancreatic sphincter, right where your common bile duct attaches to your duodenum, loosens up, and the bile then goes into the duodenum through this little hole in this mountain-like structure called your major duodenal papilla. And so now that bile is in your duodenum, so it can help in the physical digestion of fats, or the emulsification of fats within your duodenum. Okay, so now let's talk about the pancreas. So the pancreas, we said, has these little microscopic clusters of cells called acini making pancreatic juices, and the pancreatic juices contain protein and lipid digesting enzymes. So what happens is that the acini dump those pancreatic juices into little ducts, and those ducts converge and converge and converge and converge and converge until they finally converge into your main pancreatic duct, which I'm going to color in purple because it complements the green pretty well. So now we have pancreatic juices in your main pancreatic duct, also known as your duct of Wirsong. You also have an accessory pancreatic duct called your duct of Santorini, but we're not going to draw that in. It's not drawn in the diagram. But it basically does the same thing. It's just collecting pancreatic juices from all of these acini throughout the pancreas. And then finally, this main pancreatic duct, or duct of Wirsong, meets the common bile duct to form this common open space, or little lake, if you will, called the hepatopancreatic ampulla. And the common hepatopancreatic ampulla is like this little temporary mixing reservoir for the bile and the pancreatic juices. And both of these juices cannot move any further because of the occluded hepatopancreatic sphincter. So when that hepatopancreatic sphincter loosens up, the contents in that hepatopancreatic ampulla, which is the bile and the pancreatic juices, are allowed to move forward and spill out of the major duodenal papilla into the duodenum. So now we have both pancreatic juices and bile in the duodenum at the same time. Remember, we talked about the small intestine is where most of the digestion occurs. And a lot of that digestion is made possible through these two major secretions. The common, or rather not the common, but the bile itself is allowing for physical breakdown or emulsification of fats. And then the enzymes within the pancreatic juices are allowing for the actual chemical breakdown of both proteins and fats. All right, so now let's look at this diagram right here. Let's just do some more labeling. Number 4, we have our gallbladder. Number 2 represents your hepatic ducts, both the right and left hepatic ducts. So we're going to write in parentheses right and left. They converge together to form number 6, which is your common hepatic duct. And down right here, you can see where the common hepatic duct gives rise to your common bile duct, number 7. And also gives rise to number 1, which is your cystic duct. So we said that under normal circumstances, that bile goes down the common hepatic duct and then down into the common bile duct, but it gets blocked from moving any further by that hepatopancreatic sphincter, and then so it goes back up and then up through the cystic duct for temporary storage in the gallbladder. Then when the gallbladder contracts and that hepatopancreatic sphincter opens, the bile is allowed to go out through the major duodenal papilla into the duodenum so that it can assist in the emulsification or physical breakdown of fats. Number 2 is the duodenum. Number 3 is pointing towards that hepatopancreatic sphincter. And then over here on the pancreas side, number 8 is the pancreas. And then number 9 is going to be your main pancreatic duct or duct of Wirsong. All right, and that is it for this main packet, the Digestive System packet, Chapter 23. So I'm going to go through digestive physiology packet with you, and then the absorption packet. I'm probably going to touch upon the tunics very briefly as well. But you know, the tunics packet is pretty self-explanatory. We just want you to know the different layers or tunics of the alimentary canal or the GI tract. All right. So this is basically the downslide of the digestive system. You've had the vast majority of the material covered for you. Now we just have to kind of tie up some loose ends with these other packets. All right, and I will see you soon.